Hello friends, welcome. I Dr. Assistant Professor, Department of Zoology, Sharachandra Mahavidyalaya, Nodal. Friends, today we will discuss the topic of BSc Part 2nd, Semester 3rd, Zoology Paper 6 Biochemistry. In Biochemistry, Chapter 1, Nucleic acid, that is the DNA and the RNA. Friends, here the DNA and the RNA, these are nothing but the nucleic acids. Why they are called as acids? Because basically acid and base, these nucleic acids have a negative charge. So while considering the differentiation among these, they have having the different types of the characteristics. Friends, here we will differentiate the DNA and the RNA according to their structure, according to their functionality, and according to their types. As well as RNA have different types of its forms, and we will study the function one by one. So here we will start with the DNA, that is deoxyribonucleic acid. Before we study the DNA, there are the various researchers who spent their life for the invention of these nucleic acids or the DNA. So here I will tell you some scientist name, that is first one we consider Louis Pasteur, then Watson Crick, again Barbara McClintock, again Frederick Mitchell, and we consider Rosalind Franklin. So these are the few names, and the basic and the very ideal discovery in the 19th century, that is the discovery of the DNA. Here we will study the DNA, that is the deoxyribonucleic acid. It consists of the purines and the pyrimidines. There are it having the different types of the forms, for example aDNA, bDNA, zDNA. They are having the two types of the conformation, that is the right-handed and the left-handed. So while considering these conformations, we will study how they are having such type of the conformations. So while considering of the DNA, we will study historical preview of this. There are the few scientists, for example Griffith experimentation, Avery McCulloch experimentation, Hershey and Chase experimentation. In every experimentation, they determine that how the DNA is responsible for the genetic transformation. Griffith coined the term factor, but after that Avery McCulloch and McCarthy, they determined that this factor is nothing but that is responsible for the transformation of the virulent strain into the virulent strain in the case of rat. So while considering this type of the inventions, Hershey and Chase experimentation, that is the protein is not responsible for the transformation and the DNA is responsible for the transformation, that has been studied by the Hershey and Chase experimentation, or that is the ideal intervention of them. So while considering such a type of the forms, we will go toward the next, that is the invention of the DNA. Watson and Crick and Wilkinson, they got a Nobel for their discovery, that is the DNA having the double strand, and whatever may be the DNA they interpreted or they explained, that is nothing but the B form of the DNA, and that having the 10.5 base pairs. So first of all we consider the structure of the DNA. It consists of the sugar and it consists of the nucleotides. So what type of the sugar it consists of, what type of the nucleotide it consists of, and what type of the polynucleotide chain it have, that will be explained one by one. So sugar, it consists of the deoxyribose sugar, nucleotides, that is adenine, thymine, guanine, cytosine. So in spite of the thymine, in the case of RNA, uracil. Polynucleotide chain, it means that number of nucleotide component, that is they come towards and they form long chain of the nucleotide, is called as the polynucleotide chain. Polynucleotide chain plays an important role in the formation of the DNA strands. Basically they are having the anti-parallel strands, that is 5' to 3' and 3' to 5'. They are anti-parallel because they are 5' phosphate and 3' OH, they are anti-parallel towards each other, and the bonds which has been present among the adenine and thymine, that is double hydrogen bonds, and guanine and cytosine, that is triple hydrogen bonds. So they follow the Chargaff's rule. That means the percent of adenine and thymine is always equal to the guanine and cytosine. Also, DNA consists of these arms, or we consider the major and the minor group. Again, helical patterns. So these helical patterns is responsible for the twisting, and they also have the nitrogenous bases as well as the oxygen group, phosphorus group, and the hydrogen, that is, and the carbonic components. So all these components is present inside the DNA. Nucleic acid is the fourth major important biomolecule in the living organism, because this building block molecule is responsible for the genetic conformation and the structural and the functional formations. So here we consider the Watson and Crick double helical DNA molecule. After the invention of the Rosalind Franklin, that is with the help of X-ray crystallography, she interpreted that a particular DNA molecule is having the plus like sign or the X-ray crystallographic model, and according to that crystallographic model, she interpreted a particular imaginary representation, and that according to that image, afterwards Wilkinson and Watson and Crick, they determined that this model is nothing but the or this DNA model is the double stranded, having antiparallel strands consist of the purines and pyrimidines. The purines are the dicyclic and pyrimidines are the monocyclic components. They are interconnected to each other, and always the synthesis of the DNA is taking place in the 5' to 3' direction. They are having the shallow arms as well as the major and the minor grooves. So this type of the component that is responsible for the one single helical component that consists of about the 10.5 base pairs, and the DNA they interpreted that is the B form of the DNA, and that B form of the DNA is highly stable as compared to the other form of the DNA. So Watson and Crick double helical model, they interpreted that this model is the ideal model and having the such type of the patterns about 3.4 angstrom per helix, as well as the helix which consist of the nitrogenous bases as well as the phosphodiester components, as well as the double hydrogen bonds among the nitrogenous bases. They provide the stability, and this factor or this DNA is mainly responsible for the genetic transformation and the unit of the heredity. The forms of DNA, here we will study A form of the DNA, B form of the DNA, and the Z form of the DNA. B form of the DNA is highly stable, and which is present according to moisture level or the humidity of our cells. A form of the DNA is the right-handed conformational oriented DNA, and the B form of the DNA is also right-handed helical or the conformation oriented DNA. But as compared to the A form and the B form of the DNA, the Z form of the DNA is left-handed conformation oriented, or which it moves, that means to the left-handed conformation way. So that is the forms of DNA, and these forms of DNA according to different types of the metabolic metals components which has been present inside the temperature, as well as the various type of the physicochemical factors, and the humidity, moisture level is also plays an important in the occurrence of the different types of the DNA. Now we are moving toward the next part, that is a ribonucleic acid, and that is nothing but the structure of RNA. Basically, RNA is ribonucleic acid. It is a single stranded. We have the different types of the that is messenger RNA. It is also called the mRNA or the template RNA. Ribosomal RNA, it is called as the rRNA. Soluble, that is sRNA or adapter RNA or transfer RNA. So here the percentage of the mRNA is less as compared to the tRNA, and percent of the ribosomal RNA, that is more as compared to the tRNA. So this RNA having the different types of the models, that is hairpin model, chlorelic model, RNA and the DNA. Here there are the different questions arises in mind, that is whether the DNA first or the RNA first. But friends, here RNA first, because there are the different types of the evidences, because every reaction needed the catalyst or the enzyme. So this ribonucleic acid or the RNA itself acts as the ribozymes, and this ribozymes is responsible for the formation of the RNA, and after the RNA DNA formation is taking place. That's why RNA first. So here we will concentrating on the RNA. After the central dogma or the study of the central dogma of the molecular biology, we will able to determine that how the replication occurs, how the transcription occurs, how the translation occurs. So the message of the protein synthesis has been carried out by the mRNA, that is also called the template RNA. It provides this place to bind the ribosomal smaller and the larger subunit, as well as it also provides the space and to recognize the codon sequence for the tRNA. So this messenger RNA it carries the code in the form of codons. Again, ribosomal RNA having the different the ribosomes are 18S ribosomes and the 17S ribosomes. 18S ribosome present inside the eukaryotes, and the 17S ribosome which is present inside the eukaryotes, and the 70S in the prokaryotes. So while considering this ribosomal RNA, it provides the place or to bind with the particular mRNA molecule, and that sequence has been transcribed with the help of tRNA. So tRNA having the different this APE acceptor, P for the processing we consider, and the E for the exit. So while considering the transformation of the amino acids, while reading the anticodon sequences, that anticodon sequences and the transformation of the polypeptide or the formation of the amino acid, and they form the polypeptide units. So such type of the process is related with the RNA. So here we will consider the difference of this basically DNA and the RNA. So DNA, it consists of large number of the nucleotides up to 4.3 millions, and RNA which is consist of the about 12,000 nucleotides. DNA is only one type, but RNA, but it is not a case. It having the different types of the forms, but RNA having the several types. For example, messenger RNA, rRNA, and the tRNA. DNA is the usually genetic material, but RNA is also the genetic material of some viruses. So it is usually double stranded, RNA is a single stranded. There are fewer uncommon bases, and there are more common bases in the case of RNA. Genetic messengers are usually encoded in DNA, while the usual function of RNA is transcribe the message enclosed in the DNA into proteins. So during the synthesis, exonuclease is needed, and during the biosynthesis, exonuclease is normally not needed. So in the case of DNA, exonuclease is an enzyme which plays an important role in the recombination and the recombinant technology. So such a type of the exonuclease has been not needed in the case of RNA. So here again we will revise this RNA and the DNA. DNA is a double stranded, RNA is a single stranded. DNA is a participate in the genetic transformation, number of organisms, but RNA has also worked as the genetic constitution or the basically as the genome of two viruses. RNA has several types, it has different forms, it consists of the nucleotides, again it having the only one strand, it having the two strand, that is 5 prime to 3 prime, 3 prime to 5 prime. So that's all about this, and we will see the part of this syllabus one by one. Thank you, thanks once again, have a good day.